Good morning students, hope you all are fine at your home. So in the last class we have discussed about the reproduction and its type. So today we're going to deal with the asexual reproduction. Clear? So let's get started. Okay, asexual reproduction, I told you that a single parent is involved, right? So asexual reproduction have various types like binary fission, budding, fragmentation. Fourth is regeneration. Number 5, spore formation. And number 6 is vegetative propagation. So these are the different phases which comes under the asexual reproduction, and each of them we're going to discuss in detail. So talking about the binary fission. So binary fission is a type of asexual reproduction in which one organism split into two by the process of karyokinesis and cytokinesis. See, this is for example an organism. It has a nucleus. Basically, the binary fission occur in unicellular organism which have a single cell and a nucleus. So, in this organism, the division of nucleus takes place. The nucleus start dividing, and this phase is known as karyokinesis, that is nucleus division. Clear? After nucleus division, when the nucleus get divided into two, the cytoplasm start dividing. Okay, see the cytoplasmic division. So one organism split into two by the process of karyokinesis and cytokinesis. Cytokinesis is the cytoplasmic division which occurs in an organism. So here one organism separate into two daughter cell. You can see this is known as binary fission. An organism which undergoing the binary fission is amoeba. Example is amoeba. So binary fission is a process by which organism split into two equal half is known as binary fission. So binary fission example is amoeba, where one amoeba gets separated into two amoebae by the process of karyokinesis and cytokinesis. Clear? The next thing is budding. So what happens in the budding? You can see there is an organism, okay. After nuclear division, there is one small bud formation which takes place in the organism. Okay, this is the parent cell. Okay, parent cell is what? Which undergoing division is said to be parent cell. Okay, so the parent cell, the nucleus start dividing, karyokinesis takes place. So there is a bud formation. Bud, what is bud? Bud is nothing but a small outgrowth which forms on the parent cell. So nuclear division takes place, then after that the cytoplasmic division takes place. So the parent cell separate from the daughter cell. This is known as the budding. Okay, so parent cell, there is a formation of small outgrowth, nuclear division takes place, cytoplasmic division takes place, and there will be a formation of the two daughter cell. And you can see the major difference between budding and binary fission is that in binary fission it will generate the similar daughter cell. Okay? But in case of budding, one daughter cell is larger in size and one daughter cell smaller in size. You can say this is the bud one, right? Clear? So this is the basic difference between budding and binary fission. So binary fission, I have given you example amoeba. In budding, example is yeast, which is a form of fungi, and hydra. Okay, these two are the examples for the budding. Clear? So that's all about the budding. Next is fragmentation. So what happened? Fragmentation means something that is forming of the fragments. Clear? So here one organism, okay, separate into various fragments like this, and each fragment behave as a new individual. So what happened? Once an organism attain a particular length in the growth, so that fragment that fragment break down into different fragments of the smaller fragments, and that will generate the new organism. So for example, this is the Spirogyra. It is a green algae. Once it attain a particular length in size, it break down into the fragments, and each fragment behave as new individual. Clear? So example is Spirogyra. Spirogyra क्या है? It is a green algae. Next is regeneration. Okay, in regeneration what happens? Suppose this is an organism which you named as planaria. Okay, what is it? Planaria. So what happened if planaria lost its parts? Okay, this is the upper part, this is the middle part, and this is the lower part. So for example, this planaria broke into pieces. Okay, suppose this was the first piece, the head one, this is the trunk, and this is the lower abdomen. Okay, so this broke into different pieces. Each piece regenerate. It forms the lost part. For example, this part will form, I'll make it here. It will generate into new. Then this part will generate the head portion, right? And this part will generate the head and the thorax region if you talk about the regeneration. So see, the first part, second part and third part, they formed their lost part, and that is known as the regeneration. Clear? So regeneration occurs in the lower organism. Example is planaria. Example is planaria. So regeneration, how will define it? The regeneration is a process by which organism broke into pieces, and those pieces again form the lost parts. Clear? That is known as regeneration. So few of our body cells have this property, like liver cell, they can regenerate, like so, and these regeneration occurs in a special cells. Okay, the cells can form their lost parts. Next, the spore formation. Spore formation, the spores are formed by various organisms, like it being formed by bacteria, it being formed by the fungi. So what happens in spore formation? Under unfavorable condition, the organism, suppose this is a fungi and this is a fungi, this is a sporangium, the structure in which spores are formed. This is sporangiophore. This is stalk-like structure, and these are some hyphae. Okay, so the sporangium forms, a sporangium is a structure which forms spores. Clear? If it is formed inside the sporangium, they are said to be endospores, and if they are found outside the sporangium, they are said to be exospores. So these spores are formed under unfavorable condition, when there is no water or any kind of the substances which is not available to the organism for its growth. So in the absence of those facilities, it's said to be unfavorable conditions. So under unfavorable condition, the organism forms spores, and these spores are dispersed by the air currents or water current to the distant places, and when they find the suitable place for their growth, they will again germinate. This is known as sporulation or spore formation. Example is fungi and bacteria. Clear? The last one is vegetative propagation. So in vegetative propagation, what happened? This is the reproduction, asexual reproduction in case of plants. So in plants, there are various portions, like stem is there, roots are there, and leaves. So from these parts, you will get new plant. From these parts, you can get new plant. That process is known as vegetative propagation. So any of the plant part from which you can get new plant is said to be vegetative propagation. For example, if we talk about the artificial propagation, there are three processes which comes. Number one is cutting, number two is layering, and number three is grafting. If propagation is done naturally, it is said to be vegetative propagation. If it is done by the human beings, okay, that is called as artificial propagation. So from any part, like for example, we'll take about example of the rose plant. So if you cut the stem of the rose, and you bury it inside the soil and provide water and essential nutrients to the stem, you will find a new plant. You don't need seed for the growth of the plant, because from any part of the plant, if the plants have certain ability to regenerate, like sugar cane. ठीक है, शुगर केन में क्या होता है, आप उसकी स्टेम पार्ट्स को ले लेते हो, नोड इंटरनोड, इजंट इट, और फिर उनको सॉइल में डाल देते हो, यू विल गेट अ न्यू प्लांट. You don't require seed for the growth of the plant. So this is known as vegetative propagation. Clear? Like leaves, I'll give you the example, bryophyllum. Bryophyllum. These plant have the leaves of this plant will fall onto the ground, and they find suitable water and nutrients. Then again they will form the roots. Like suppose this is the ground and this is the leaf of the bryophyllum. Where it is touched the ground, it start forming the roots. Clear? And from this a new plant arises. This is the example of the leaf in vegetative propagation. Clear? For the stem you can know very well the rose, and for root you can give example of the dahlia. Right? So these are some examples for the vegetative propagation. Next is the artificial propagation. I told you this is man-made. So cutting, I'll give you the example of rose. Again, if you cut the stem of the particular plant and bury it inside the soil, you will get new plant. That is cutting. What is layering? Okay, suppose this is a jasmine plant. Okay, these are the branches of the jasmine plant. These are the lower branches, right? If you bend these branches and bury it inside the soil without breaking it, so after few days you will find some kind of rooting, right? And then you cut the stem from here, so you got a new plant from the main plant. Like this is known as layering. Clear? This procedure is known as layering. The next thing is grafting, and this occurs in the, suppose the cutting occurs in rose and this occurs in the jasmine, the layering. And the next is grafting. Suppose there are two plants, okay? I named it A. The second one is B. I'll take the example of oranges. A plant, basically, okay, it produces more fruits. The orange are produced by this plant is more in number. Clear? So more fruits are produced by the plant A. And what kind of fruits? Oranges. The B plant produces less fruits, for example. And these more fruits have more number of seeds in them. If you eat oranges, you find seeds, right? So A plant produces more fruits, but what is the drawback? There are more seed in those oranges. The next thing is they no seed. This plant produces less fruit or less oranges, but there are no seeds in these oranges. It's a miracle, isn't it? But I want a property in a plant that I generate more fruits in that plant and without seed. So what I will do, I will, suppose this is my plant A and this is my plant B. I told you it produces more fruits and it produces less fruits, है ना? But there are no seeds in this B1. Here more seeds. So I want quality that my plant should generate more fruits without seed. So what I will do, I will take the grafts from these plants. What do you mean by graft? I'll make a cut like this, like, okay? So slanting cut. So this portion of the plant without root is said to be scion, and this portion with roots of the plant is known as stalk. Clear? So this portion is known as scion without roots and this portion is known as stalk. So I will take the scion of plant B and take stalk of plant A. So I'll take stalk of plant A, which is the part with the root, and I'll take the scion part of plant B. Okay, see, this technique of grafting is generally done in dicot plants because there is a cambium layer. Clear? So these technique is done only in the dicot plants, not in monocot plants. So I'll take the scion of B and stalk of A. Then I will join these grafts with the help of cellophane or polythene cover. Clear? So that there is no infection in those plants. So after some time you will find the proper plant, and this technique is known as grafting. Clear? This technique is known as grafting. So this procedure will give me two results. This procedure will give me two results. Maybe I'll get less fruits with the more seeds, or I'll get more fruits with without seed. So there is a chances of both the procedure or both the properties to come in the plant. So that's all about the vegetative propagation, where I have explained you about the artificial vegetative propagation and vegetative propagation, where other the examples of the asexual reproduction. Clear? In the next class we will discuss about the sexual reproduction in human beings. Thank you.